In this video, we're going to be looking at transport in plants, and this is going to finish off section 3 of our curriculum. So first of all, the learning questions. So by the end of this video, you should be able to write up answers to explain the role of the xylem in plants, explain how water transport in the xylem is linked to transpiration in the leaves, explain the cohesion and transport, explain the factors which affect transpiration, explain the role of the phloem in plants, explain what is meant by translocation, and explain the mass flow hypothesis as a mechanism for translocation. Theory of water transport. So looking at why do multicellular plants need transport systems? Okay, well, first of all, the metabolic demands of a plant. Okay, so the internal and underground parts of a plant do not photosynthesize, and this is because obviously there is no sunlight available under the ground, but they will still require oxygen and glucose to be transported to them, and it will also require the waste products of cell metabolism to be removed. Hormones made in one area of a plant need to be transported to another area to carry out an effect, and mineral ions absorbed by the roots from the soil need to be transported to all cells to make the proteins required for enzymes and the structure of the cell. The next is the size. So plants need an effective system to move substances both up and down from the tip of the roots to the topmost leaves and stems, and transport substances against the force of gravity. And these can be extensive lengths. So if you think about the size of trees, they could be up to 50 meters in height. Okay, looking at the surface area to volume ratio. So the size and the complexity of the multicellular plant means that they actually have a relatively small surface area to volume ratio when you take into account all parts of the plant, the stem, trunks and roots. Therefore, we cannot rely on diffusion alone to supply the cells with everything they need. So looking at the structure and function of the vascular systems in roots, stems and leaves of herbaceous and dicotyledonous plants. Now, this is not required by AQA, but it is something that will come up often in exam questions, so it's important to be able to understand and familiarize ourselves with these words. So dicotyledonous plants are plants that contain two cotyledons, and cotyledons are the organs that act as food stores for the developing embryo and form the first leaves when the seed germinates. Herbaceous dicots are soft tissued and have short life cycles, and woody dicots have hard lignified tissue and a long life cycle. The vascular system, which just is comprised of our xylem and phloem, our transport tissues, are arranged in vascular bundles. So looking at the structure of the vascular bundles in the roots, so they're going to have a drill-like structure, okay? So you can see the xylem is in the center, and the xylem is the strongest tissue, and it provides some mechanical strength. And this is quite important, as they're going to have to be able to withstand pressure, pulling pressure from the stem and the leaves, from wind, and also from people. Okay, as people run through them or as animals run through them, they may catch onto the stem and pull, and that root is what anchors the plant. Okay, so in the endodermis around the vascular bundle, there is a layer of meristem cells that can form the pericycle. Now remember, the meristem cells are cells that contain undifferentiated cells. Okay, and this is really important. That drill-like structure is enabling the plant to push down into the root. Okay. Now looking next at the vascular bundles in the stem. So the xylem is located on the inside, and this again is adding support to the stem. We have our parenchyma, which is found in the center of the vessel, and this is just a packing tissue. Okay. That cambium layer between the xylem and the phloem is again our meristem cells, which can produce new xylem and phloem cells tissue when required. The vascular bundles, as they're located around the edge, they give the stem support and strength. Looking next at the leaf, okay, so this is in the midrib. Okay, and the midrib is the main vein. So if you ever look at a leaf, it has that vein structure. Okay, so we have our midrib, and there's going to be veins that branch off it. Okay, and as you can see, the xylem is located on top of the phloem. Again, this is to provide support. Okay, that midrib carrying the vascular tissue helps support the leaf and keep the leaf upright. Okay, and in doing so, it allows it to capture sunlight and carry out photosynthesis. Okay, so if you're given the transverse section, so TS means transverse section. Now, if you're given a transverse section of the different parts or of the different vascular bundles in different parts of the plants, just have a look at what they look like. So remember, in the stem, they're located around the edge to give strength and support. In a root, they're located in the center to withstand tugging strains. And in the leaf, they're in the main vein. And this is going to support the structure of the leaf. And then again, we have the many branching veins off that midrib, aiding in transport and support also. Looking next at the structure of xylem and how this structure relates to its function. So the function of xylem is the transport of water and mineral ions and also to support the plant. It's made up of non-living tissue, and it's formed by long continuous columns made of dead tissue that are open-ended, and this allows the transportation of water. Now, this long continuous columns contain pits, and this pits allows water to move laterally between vessels. It's thickened with tough substance called lignin, and this is deposited in a spiral pattern. Okay, and this is important as it allows the plant to remain flexible, but also reinforces the lignin so that it does not collapse under the transpiration pull, i.e. as water is pulled through it or forced. The thick wall parenchyma packs around the vessels, and this stores food in tannin deposits. Now, these tannin deposits are what is found, we know, after a cup of tea, there's rings around the cup. These are chemicals, and they're bitter and astringent tasting, and they protect the plant tissue from any attack by herbivores. So they act as a deterrent almost. So here we have the structure of the xylem vessel, and we can see that it's a long continuous column, and this column is hollow. Okay, it has no cytoplasm, it's a dead cell. Okay, it has a lignified cell wall for support. That parenchyma is our packing tissue, and the spiral of lignin are going to help reinforce the xylem vessels so that they do not collapse under the transpiration pull. Looking now at the structure and function of phloem. So phloem is a living tissue that's job is to transport nutrients to storage organs and also to growing parts of the plant. And this transport of nutrients around the plant is called translocation. Nutrients are dissolved in the form of sap, and this movement can be upwards or downwards. The main transporting vessels are our sieve tube elements, and this is when many cells joined end to end form a long hollow structure, and there are sieve plates between these areas, and this allows sap to pass through. Companion cells are linked to the sieve tube elements by plasmodesmata, and these plasmodesmata are just channels that link one sieve tube element to another cytoplasm within them, and it is channeled through the cellulose wall. And the role of companion cell is that it contains many mitochondria, allowing the active transport of sugar into those sieve tube elements. Okay. So here we can see the perforated sieve plate that allows sap to pass through, the companion cells, which is going to contain a lot of mitochondria so that active transport can be used to actively pump the solids around. And also you can see here that there's a cytoplasm because the phloem is a living tissue. Okay. So looking at the process of transpiration and the environmental factors that will affect the transpiration rate. So looking at why water is important in plants. Well, first of all, turgor pressure provides a hydrostatic skeleton that's going to support the stem and leaves. This turgor drives cell expansion. So often you see buckled pavements, and this is because plant roots are going to force their way through substances such as tarmac when they grow. Loss of water by evaporation is going to help the plant cool. Mineral ions and products of photosynthesis are transported in aqueous solutions. So that means that our minerals and nutrients are dissolved in water. And water is also a raw material or a reactant for photosynthesis. Okay, looking and thinking back to our key stage 4, the adaptations of root hairs. So their role is to absorb water by osmosis. So looking at that, how are they adapted to do their job? So they have a microscopic size, meaning they can easily penetrate through soil particles, and this allows them to gain access to even more water. Having a large surface area to volume ratio, there's also thousands of root hairs on each growing root tip. Now, we have often talked about, whenever we talk about exchange surfaces, we need to discuss the surface area to volume ratio. We talk about diffusion gradient and diffusion distance. And here we're just going to do exactly that. So each hair has a thin surface layer. That means that the distance for diffusion is short, and also the distance for osmosis is short too. The concentration of solutes in the cytoplasm of root hair cells is maintaining that water potential gradient between the soil water and the cell. Okay? Looking at how water enters the root hair cell. So water is going to enter the root hair cell and move into the xylem tissue location, the centre of the root. And this movement is going to occur as a result of the water potential gradient, as the water potential is higher inside the soil than inside the root hair cell. And this is due to the dissolved substances in the cell sap, which are going to lower the water potential. Therefore, the purpose of root hair cells is to provide a large surface area for the movement of water to occur. Minerals are also absorbed through the root hair cells by AQA Transport, as they need to be pumped against the concentration gradient. And we said that this is going to require energy, thus the presence of mitochondria is important. Okay, so there's two ways water can be taken up by the root and taken to the xylem. And this is called, it can occur either via the symplast pathway or the apoplast pathway. So first of all, the symplast pathway is when it travels through the cytoplasm. Okay, so water is going to enter the cytoplasm and it's going to pass from one cell to the next via the plasmodesmata, and all it's moving is from high water potential to low water potential, as each adjacent cell as it moves to is going to have a lower water potential than the cell it just came from. The other pathway is the apoplast pathway, which is through the cell wall or through the intracellular spaces. So it's going to move through the water-filled spaces between the cellulose molecules in the cell walls. In this way, water doesn't pass through any plasma membranes, therefore it can carry dissolved mineral ions and salts. When the water reaches a part of the root called the endodermis, it reaches this layer of sebarin which cannot allow water pass through, and this is called the Casparian strip, which we can see here is that black mark. Okay, so water cannot pass through, and therefore water is going to have to move this into the symplast pathway. So it's going to be moved into the cytoplasm of the root hair cell. So once it has passed through the endodermis, it can continue down the water potential gradient from cell to cell until it reaches a pit in the xylem vessel, which is now its entry point for water. Okay, so looking at the transport of water through the leaf and to the air surrounding the leaves. Now, this whole process is in essence transpiration, the loss of water vapour from leaves and stems of plants. It's a consequence of gaseous exchange. And this is why I say this is because it's going to occur when the plant opens the stomata to exchange oxygen and carbon dioxide with the atmosphere. So looking at the transpiration stream. The transpiration stream is the movement of water from its uptake in the roots to its losses in the leaves. The water molecules evaporate from the surface of the mesophyll cell, then move through the air spaces in the leaf and out of the stomata by diffusion. The loss of water lowers the water potential, so water moves from the neighbouring cell into the cell, the water is just evaporated from by osmosis. And this is going to be repeated all the way back to the xylem. Now, looking at how this water is going to form this continuous stream that's pulled along. Well, first of all, there's adhesion, and this is when water molecules form bonds with walls of xylem vessels. And also cohesion. Okay, as water is a polar molecule, there's going to be hydrogen bonding formed with other water molecules. And this results in water being pulled up the xylem vessel in a phenomenon called capillary action. Capillary action describes when water is going to move up a narrow tube against the force of gravity. Looking at the transpiration pull, it's the continuous movement of water up a plant to replace water lost by evaporation. And this is going to result in a tension, which in turn helps the water move across the roots from the soil. Okay, so we can see that transpiration involves osmosis, where water moves from the xylem to the mesophyll cells, and it also involves evaporation from the surface of mesophyll cells into the intracellular spaces, and the diffusion of water vapour down a water vapour potential gradient out of the stomata. Okay, so we can see here water is drawn in the transpiration stream as a result of the cohesive forces between water molecules. As this water evaporates from the surface of the mesophyll cell, it's going to leave its water down the water vapour potential gradient into the intracellular spaces. Okay, and we can see here that this is going to create tension the whole way down along towards the root. Okay, looking at, well, how do we know this is true? What evidence do we have for the cohesion tension theory? Well, if there's changes in the diameters of trees and transpiration is at its highest during the day, so the xylem vessels are going to be at its greatest tension, so the water shrinks in the tree shrinks, in diameter. When a xylem vessel is broken, air is going to be drawn in rather than leaking out, and no water can be pulled up the xylem vessel as the chain of cohesive forces is now broken. Okay, so if we want to measure transpiration, we can use a potometer where water lost by the leaf is going to be replaced by water in a capillary tube, and therefore measuring the movement of the meniscus or the bubble, we can determine the rate of transpiration. Okay, so the potometer is an apparatus which creates a sealed environment with water, which can be used to measure water uptake based on how far that air-water meniscus moves over time. Now, in an exam question situation, you must understand that this apparatus must be assembled under water and sealed with waterproof jelly, like Vaseline, to keep it airtight, because, as we discussed earlier, any water that gets in is going to break the transpiration stream. It's going to interrupt the cohesive forces, therefore stopping the transpiration pull. Okay, looking at how stomata control the rate of transpiration. So we said that the opening and closing of stomata is a turgor-driven process. It relies on the movement of water into and out of the cell, creating a turgid cell, or thus, if water leaves, creating a flaccid cell. When turgor is low, or hormone signals from the root, the stomata will close. Now this is quite clever, because if in a period of drought, hormones from the root soil will be carried to, hence the requirements for a specialised transport system, to the stomata. It's going to signify for them to close. If the environmental conditions are favourable, the guard cells are going to pump in solutes back to transport, and this is going to increase turgor, allowing stomata to open, allowing the transpiration stream to continue. Okay, looking at the factors that affect transpiration. So it's important, the factors that affect transpiration are going to include the number of leaves, number, size, or the position of the stomata, the presence of the waxy cuticle, the amount of light presence, the temperature, humidity of the air, and the air movement and water availability. So more light means that more photosynthesis is happening. Therefore, more stomata open as the light intensity increases because more oxygen is required. However, if the stomata are open, more water is going to leave, so transpiration is going to increase. Looking at next, the more humid the environment, the lower the water potential gradient between the inside and the outside of the plant, so less transpiration as diffusion out is slower. Now remember, humidity is a measure of the water content of the air. However, we can also create humid microclimates, and these can be created with pitted stomata, hairy leaves or rolled leaves, and they're going to basically reduce the water potential gradient right outside the stomata, thus reducing the rate of transpiration. Increase in temperature is going to increase the rate of evaporation as the kinetic energy, apologies, there should be an I in kinetic energy, as the rate of kinetic energy of the water molecules are going to increase. Thus, this is going to increase the rate of the movement of water from the spongy mesophyll cells into the air spaces of the leaf. Okay, so that sentence is quite poorly written. Increases the rate of kinetic energy of the water molecules, thus increasing the rate of evaporation, and that's from the spongy mesophyll into the air spaces of the leaf. Increase in temperature is also going to increase the amount of water the outside air can hold, thus further increasing the diffusion gradient. The more water available, the greater the rate of transpiration as more is taken up by the roots. And the air movement. So the microenvironment of water vapour around the stomata, so there's this still microclimate of air, and the leaf surface is blown away. And this is going to reduce the water vapour potential around the stomata, which is going to increase the potential gradient. Okay, so we can see here there's a microclimate of still air around the stomata, which is decreasing the water vapour potential gradient. If a strong wind comes and interrupts that microclimate, the water potential gradient is going to be increased, thus the rate of transpiration will increase.